Yes. Hello, everyone. Thank you for coming. So it's interesting because almost every time we have new people coming, it means that people interested in different topics. Hence, I have to first say what we are doing for those who came here for the first time. We are giving cycles of lectures, which are called Adventures through the Laws of Nature. And we try to go through all the most fundamental things we know about the nature. So we went through all corners of physics already. And today we turn a little bit around and come back to composition of different physical laws to understand how life is going. And today we have pleasure, Philip Mai is going to tell us about biophysics. I hope it will be very different and interesting experience compared to boring theoretical physics guys speaking about it. We'll have a lot of things to do. So please, Philip.
Thank you very much. And I don't think I will disappoint in the sense of being very different. It will definitely be very different. I think it will be much more messy and not as elegant as the lectures that you've seen until now. This is more of a mixture of almost engineering with physics and biology to try to understand the natural world. And I've entitled my lecture today, Seeing the Invisible and the Structure of Life, because that's been a challenge throughout, I would say, humanity, trying to understand how things work. And it's still a mystery in many ways how life works. We know a lot, but there's still a lot that we don't know. And you might, or I put a structure or a sculpture actually on the top of my first slide. Can anyone tell me what do you think that structure represents? DNA, exactly. So DNA was maybe one of those great achievements in terms of biology, in terms of being able to unify the messiness that is biology, when people were able to find out that DNA pretty much exists everywhere on all living things. Maybe not always exactly DNA. I mean, you have some viruses which you might say maybe are not really alive, which might have RNA. But in any case, you have nucleic acid somewhere in everywhere. While if you just look from the point of view of microscopic world, living things look completely different. I mean, you have people, we have trees, you have bacteria and so on, and they look nothing alike, but yet they share many very fundamental things. It's just that they're not immediately apparent to our naked eye. But the best observation instrument that we have, I think, is without a doubt just our eyes and our ability to look around and see things. And it's been an amazing instrument, and it's still the one we use the most. But it has some limitations. Like, for example, we cannot just look at something and see its DNA because our eyes have their limitations, even though they're really, really good. And so people have tried through thousands of years, actually, come up with ways to improve how our senses and our abilities to see things. So I have one example here that I found, and this was actually probably not made for improving the eyes directly. It might have been, you have this 3,000-year-old lens made out of stone, and this was discovered in the Middle East. And it's not entirely clear what this was used for. It might have been, for example, just to try to start fires using sunlight or some other reason, but it's obvious that they were trying to do some kind of optics. So optics in this sense goes back a really long way. And the whole point is just that we're able to try to increase the capacity of our eyes. And here you have maybe one of the most simple tools that you could use, simply a magnifying lens to try to look at things in more detail. And you can use this to already get some amount of magnification, but up to where can you get using this type of tricks? Well, if you're just using a magnifying lens, not very far. But it has been known for a very long time that the basic way that lenses work is in this simple geometric optics approach, you just have, for example, in this case, parallel lights of ray incident on a lens, and then those, if you have a very good lens, they will, or if you have a good lens, and you have monochromatic light, for example, you will be able to focus all your light in a single point, and then you should be able to obtain then high quality pictures in this simple formulation. And this is just a picture showing this in practice, where you have 3 light rays and they all kind of converge into one point. And this is just to show you that these lenses can be used to magnify. And it is not clear from just these pictures what are the limits that you might have. And this has been used for a very long time, lenses. So here we have an example from the 17th century from Galileo, who was maybe more famous for building telescopes, but among other things in terms of the in terms of the instrumentation building. But he also built actually some of the first microscopes, because the technologies are actually very related. And this is just an example of one of the microscopes. I think this is a replica of something that he built. And one of maybe the first person to really make use of microscopes to interrogate and explore more the biological world was Robert Hooke, who is known for many things, including Hooke's law. But in here, we're more interested in looking at, I'll try to hide this thing.
Just remove here.
Is it here or?
Well, no, maybe on this feature panel.
No, that was the wrong. Yeah, this one. Okay, that's almost. So here we have a picture of Hooke's microscope setup that he used around in the 1600s to look at the world around him. And you can see that it doesn't look like a very sophisticated microscope compared to the ones we have nowadays. But you can also see here on the left-hand side that he has some candle as his light source. He couldn't really have anything much brighter at that time, and he used some lens here to try to condense, to focus the light onto his specimen, so he can have enough illumination to be able to see something from his relatively tiny aperture that he has on the bottom of his microscope. But even with this, I would say, rather primitive setup, but very advanced on the 1600s, he was able to discover some amazing things, and he wrote a book which was a real bestseller in the 1600s. Everybody had it. Everybody had to have Micrographia. And because the things that he drew were extremely interesting. So he was not only good at finding interesting things to look at, but he was very good also at drawing the things that he saw. So you might be familiar with this picture. This is a picture of a flea that is maybe one of the most famous pictures of the time. And the interesting thing here was that a flea was something that everybody was familiar with. Unfortunately, the hygienic conditions were not that great in the 1600s, so fleas were pretty common. But nobody could see a flea with this level of detail that he was able to see with his microscope. And so everybody found it really interesting to see all of these things that were crawling around, and we couldn't really understand them, what they were really doing. And here you could see everything in great detail. But maybe from a scientific point of view, there are other even more interesting things that he looked at. And this picture does not look anywhere near as spectacular as the picture of the flea that I've shown before. But so this is just a picture of some cork. So he just took some cork from a tree, and then he looked at it, and he saw these very interesting things. And to him, it looked like these were these cells, which for him, cells corresponded to the cells where monks lived in a monastery. And so they were small, like rooms next to each other. And so he decided to call these also cells. And that was pretty much the first observation of cells, and then this gave rise to a whole theory of cells. And nowadays we know that pretty much all living things are made up of cells. And he was able to observe these things already in the 1600s. So it is quite amazing how, and again, something that you couldn't really see by your naked eye, but by the use of some clever instrumentation, we were able to discover these things. And furthermore, a couple of years later, Antoine van Leeuwenhoek, who is probably also one of the people who is more closely associated with microscopes, because he was able to improve quite a lot the quality of microscopes, even though he had a rather different type of microscope design. You can see here on the top left, there's a replica. This is not an original, it's a replica of one of the microscopes that he built. And he had a magnification power of about 300 times. And with it, he could see a whole bunch of things. So not only could he see a lot of cells, so here on the top right, this is just a piece of, so just some droplets of blood that were dried out on some piece of glass. And then he looked at it and he could see red blood cells, and lots of them. Which actually at the time when he did this, they were already known, so it was not a new thing. But he was able to do this with quite high quality. And this picture here is actually taken from a microscope, which is a replica of the original microscope that he did. So the quality is expected to be similar to the quality that he was able to achieve. But more interestingly, here on the bottom left, we actually have the first picture, or Leeuwenhoek at the time was able to, for the first time, observe bacteria, which in this particular case are some bacteria that are rather long. So they're many microns in length. And you can see they have this shape, which is, they're not very fat, but they're pretty long. And so that was why he was able to see them. That was the first observation of a bacteria. And here on the right-hand side, we have actually something called a giardia, which is a parasite for us, which he also observed at the time, but these are much bigger and so easier to observe. So we were discovering all kinds of things at the time, but still things, we're still seeing a lot of diversity. It was hard to unify things and see what are the common things among all living things. I mean, we could see that we have cells pretty much everywhere, but in terms of a more fundamental level of how things really work, we still don't have, at this time, did not have that much knowledge. Furthermore, we had a bit of a complication. So it was known from people looking at stars that while a star is a really tiny, tiny thing, and you would expect from your simple ray optics understanding that to get just a really small dot, that a star would cause a small dot on an image because it has such a small angular spread because it's so far away. And even though it's quite big, the angular size is very small. In practice, what people saw was that stars, particularly bright stars, would not dot, but it would show up as we have here. There's several kinds of blobby-looking things with a certain amount of blurring, and this was a bit concerning because it limited the quality of the images that it was possible to achieve. And throughout the times, there were competing theories about light. So some people thought lights were particles, other people thought lights were waves, and this went on for a very long time. And the explanations for this came more from the side of those that thought that light were waves. And actually, it took rather long time until someone came up with an appropriate description and modeling of this distortion that you see, or this blurring that you see. George Airy in the 1800s came up with an accurate description where he was able to figure out that the amount of the size of which is called nowadays this Airy disk depends on the size of the optics that you have and the wavelength that that you're using to observe. So the larger your so-called aperture, or the larger the lens that you're able to collect light, the smaller is going to be this disk, and also the smaller the wavelength, the smaller this is going to be. And this is just to show you here a couple of examples on a more modern microscope. So if you have light which is incident just in a small region, then you will end up with an Airy disk which is going to be pretty broad if this is from a point source. While if you, as you start to increase the aperture size, or the area of the lens that collects light, you're going to end up with smaller and smaller, sharper and sharper Airy disks, and so better and better resolution. And resolution is often, there are many ways to characterize resolution. One way is, for example, to see up to what distance you can distinguish 2 point sources. And the aim here is to be able to make sure that the Airy disks from those 2 point sources do not overlap in such a way that you're not able to distinguish whether you have one single point source, or we have 2 point sources. So the conclusion, though, is that, okay, we understand that we have these Airy disks that come out from point sources, but it also kind of puts a limit on how much resolution we're able to, even if we have the most amazing optics and the most brilliant microscope builders, there's going to be a limit up to what resolution we're able to see things when we're using light, because it just there's a maximum amount of angle here that we're able to collect light from, and so there's a fundamental limit here. Fortunately, Maxwell in the 1800s came up with this wonderful theory of electromagnetism, which was really a wonderful achievement. And he predicted that light is not limited to the visible range, but there's also light outside of the visible range. And this was, I think, quite revolutionary at the time. And many people were somewhat skeptical. There were many people at the time trying to prove that he was also, I mean, many people were also very, they thought the idea was very appealing, and they tried to prove that there were other forms of light. And the first actual observation of light outside of the visible range was done by Hertz, and he actually discovered radio waves which had a wavelength much longer than normal light. And he did this in the end of the 1800s, and you can see here he used this very interesting instrumentation. This was actually his emitter. So there was a small spark here in the middle that caused waves to be generated, and he had a receiver here where when the waves were received, there was again a spark there's a tiny gap here on the bottom where there were some sparks again that he was able to observe. And through the instrumentation, he was able to observe radio waves. And so this discovery was amazing because it proved that Maxwell was correct. And even though we couldn't see any light at all, there was clearly some electromagnetic radiation going from the emitter to the receiver. But to him, he thought that this is absolutely of no use whatsoever. It's just an experiment that proves that the amazing Maxwell is correct. And there is all of this electromagnetic spectrum. So we have these mysterious electromagnetic waves that we cannot see with the naked eye, but they are there. And this is probably one of the times where Hertz is wrong. As you might imagine, there's plenty of uses for radio waves, like in radios or televisions or cell phones and so on and so on. But this is not what this lecture is about. So we'll quickly divert back to the rest of our lecture. So the radio waves are very interesting, but unfortunately, they're not so useful for our particular application. Was we were interested in trying to understand biology and trying to see things at a more microscopical level, more than we could achieve with a microscope. And in the end of the 1800s, cathode ray tubes were all the rage. So everybody had one of these at home, and everybody wanted to play with these things. And so cathode ray tubes, you have here some pictures on the top when it's not working, and on the bottom when you turn them on. So this is a time when everybody was trying to play around with electricity and what you can do with this. So this is just a vacuum tube where you have a glass tube and you just remove as much gas as you can. And then you apply quite a bit of voltage here between the cathode and the anode. And then there were these mysterious cathode rays at the time that people are trying to understand. And they would fly from the cathode kind of more or less straight, and they would cause this beautiful fluorescence, and they were easily absorbed by whatever you put in front of them. So in this case, some people just put a piece of cardboard, and then you were able to see that you create a shadow on the fluorescence. And people are very interested by these cathode ray tubes, and they played around with changing the pressures, and then you got different types of fluorescence. So you can see here you have some green, and then you have some blue, and so on. And it was, yeah, people are really interested in these things. And one of the reasons why this was possible was that at this time also the development of vacuum pumps improved, and you were able to get better vacuum, because if you don't have very good vacuum, then none of this happens, and it's all very boring. But as you get better and better vacuum, you're able to see more things. And cathode ray tubes, you might be more familiar, as this kind of is pretty much the same type of cathode ray tube, just the modern iteration. But it is the same thing in the old type of TVs. So from all of these experiments in the cathode ray tubes, they were actually very productive in the end. And one of the first things that were discovered was by J.J. Thomson, with many other people actually helping out. He realized, and actually other people also realized, that the cathode rays, that they didn't exactly know what they were at the time, you could put a magnet, as you can see here on top, next to them, and they would deflect the shadow, which they assume that what's going to happen is that you're deflecting whatever these cathode rays are. So he was able then to estimate in the end of the 1800s the mass of what then became called the electron, and figured out that what was going on is that the electrons these cathode rays were actually just rays of electrons. And also he was able to estimate the ratio between the mass and the charge, and then with the help of Millikan, and who was able to determine the electron charge, they were able to figure out all of these quantities, and they were actually quite amazed by how light and how fast the electron was. And he received the Nobel Prize at the time. And you will see that the electrons will come up later in lecture on as one of the possibilities to image things, but at this time it was still far out of the idea of anyone to try to use them for imaging. But again, at the same time, pretty much around the end of the 1800s, like I said, everybody was playing with these vacuum tubes, or these cathode ray tubes, including Röntgen here. So he was playing around with them, and he happened to have some fluorescent screen somewhere stored away in his lab. And he was amazed that he was able to see that they've been exposed, these fluorescent screens, even though they were stored away in some kind of wooden box. And then he repeated more experiments, and he was convinced that this was actually what had happened. Somehow the cathode rays had created some kind of other rays, which he called the X-rays, that were able to expose the fluorescent screens which were stored away. So they were able to go through wood, which was completely different from the cathode rays, which were easily blocked even by a little bit of cardboard. And this was actually great news, and he received rather quickly a Nobel Prize, the first ever Nobel Prize in 1901. And you can see here just his lab where he did his experiments. And he rapidly decided that he could use these rays, these X-rays, to take pictures of people and look inside people. And here on the left is a picture of the hand of his wife. And on the right is a picture of the hand of one of professor that he took during a public lecture. So this was, at the time, as you might imagine, being able to see the bones of living people caused quite a stir. And it was actually worldwide news rather quickly. This is a newspaper from just a couple of months after he did the initial announcement from San Francisco. So he was in Germany, and this was first published in Germany and Austria, but it spread around the world because it was so starking, these pictures, and you have these cathode ray wonders. So these rays that are coming out, waves with a new wiggle. So it's an article in the San Francisco Call newspaper at the time. So this was causing quite a stir, and people just decided to start to take X-rays out of everything they could see. So this is an example here of a chameleon from 1896. So again, just a couple of months after the discovery, it had already spread very widely, which is not common for a new scientific invention. But this was so obvious, its power, that people are using it. And actually, it got to the point where people wanted to take X-rays for fun. So this is the National Electrical Exposition in New York. This is also 1896. And this was actually, Thomas Edison built this machine to allow people to just take pictures of their hand. You just go there, there was a queue. This was just a kind of a fair that people would go there, put their hand, and then were able to see the X-ray from their hand on a fluorescent screen. And then you just go to the next one and so on. So it was very curious. And you can understand that, wow, I want to be able to see the interior of my body. This is really cool. So we're going to go and use these new X-rays to be able to do this. And yeah, people are curious, so of course they will do it. At the same time, there were also some people that were afraid that Röntgen, with his new magic camera, is going to be able to see all of the dark secrets that are hidden inside of everyone and be able to read their thoughts and all kinds of crazy things, because he had these new X-rays that were able to do. It was a bit magical at the time. So if you could see through things, maybe you can also see the thoughts of people, and there was nowhere you could hide, because these X-rays were able to see through everything. So there was a little bit of a about, yeah, you cannot hide anymore. The X-rays will see through it. So the X-rays were discovered, and it had been known for a rather long time that if you had light, which for Thomas Young it was clearly a wave, he made this wonderful experiment where he shone light through two narrow slits, and then you observe this interference pattern at a distance. And it was already known that this interference pattern depends on the wavelength of the light, and the spacing here between these lines will depend on the wavelength of light, and on the spacing between on the structure here of the slits. Another question that was totally unclear at the time was, what the hell were these X-rays? So some people thought that X-rays were a new type of particle. Other people thought that X-rays were light of a different wavelength. But there were actually quite a few attempts at trying to prove that X-ray was light, but for example, doing this kind of experiment, doing double slit experiments, and people tried quite a lot, very carefully and so on, and they were not able to see any kind of these fringes. So they were not convinced that X-ray had anything to do with light, and maybe they thought it was just something different. And that was until Maxwell Laue, he had this, I would say, quite spectacular insight that he thought that first, he was convinced that X-rays were just had a really, really tiny wavelength, and that's why people were not able to see this fringe patterns when you do these double slit experiments. So that was the first thing. And the second thing was that he thought that these wavelengths from these X-rays was probably comparable, sorry, to the atomic distance between atoms, which again at the time was it was the idea of the atoms were more of a theory than something that had been confirmed. And another third idea that he had, and again it was not clear that this was the case, was that a crystal was a regular arrangement of atoms with a clear geometry. And people had been studying crystals for a very long time, particularly those crystals that appear in nature. And they knew that there were these special angles and so on that crystals make, and special ratios between the lengths of crystals. So that was why there was this theory that crystals have a regular arrangement of atoms. And von Laue's prediction was that if this is all true, then I might be able to shine my X-rays onto a crystal and obtain a diffraction pattern, or a kind of thing that looks a little bit like a double slit experiment. I would be able to change the direction of the X-rays and have the X-rays going somewhere else, very much like you can do with the double slit experiment. And he did his first experiment on this copper sulfate crystal, or something like this, because it was easy to grow. And he got this rather ugly looking picture, but at the time, this was very, very important because it proved that the X-rays were some kind of electromagnetic wave. And this was in 1912. So a couple of years, you can see it took quite a while between X-rays were discovered and until someone was able to prove they were connected to light. It took a very long time. And then he got the Nobel Prize in 1914. So this was the first crystal diffraction. But it was quite ugly. And he kind of improved his instrumentation by improving the collimation of the X-rays, so getting the X-rays going a bit more straight and putting the detector in a better position and getting a different kind of crystal. So then he took this much nicer looking diffraction pattern from or forgiving geometry or symmetry of the crystal. So you can see this crystal is very clearly kind of like cubic looking. It's not entirely cubic, but it's all its angles are 90 degrees at least. And you can see that this diffraction pattern here, you can also see that it looks like a squarish looking thing. So there is this relationship between the diffraction or the position of the spots that you get in the diffraction and the symmetry of the crystal. So there were these beautiful patterns from Laue. Unfortunately, he couldn't really work out what was the relationship between the patterns he was getting and the structure that gave rise to the structure of the crystal. But he knew that there had to be a relationship, very much like when there is a relationship between the position of the slits and the wavelength and the double slit pattern that you get. So he worked on it for quite a while and got nowhere. But luckily, there were some father and son pair, the Braggs, who got some. So he was, I think, one of them was visiting Europe, and they were able to get a picture from one of these diffraction patterns. And William Bragg, the son, was he had studied mathematics, so he was quite familiar in a lot of mathematical theory. and then he also studied some physics. And Henry Bragg had been doing a lot of experiments with X-rays, so they had the right background. And what they figured out was a kind of a simple scheme to try to analyze the spots that you had. And what they did was they imagined that the crystal has a bunch of different layers of atoms, and here they're represented just by these black dots, each atom, as they are part of a crystal. And your X-rays, they come from a certain direction, and then they're going to be scattered in another direction. And here we'll have a certain distance between the different layers of atoms in our crystal, and we're going to have some kind of angle also made between the face of the crystal and the direction that the X-rays are coming. Okay, and what he was able to figure out is that depending on the wavelength and the angle that your X-rays come in, there are multiple alternatives that can happen. So you can have something like you have here on the left-hand side, where due to the geometry, the path length from the rays that diffract from the top layer of atoms is a multiple of the wavelength shorter than the path length of the rays that diffract from the next layer of atoms. So if you combine here this part here in red and the part here in green, you will end up, if you end up with an integer multiple of the wavelength that you're using, you will see that while you can see here that these two waves are in phase because their minima and their maxima coincide, but even after they get reflected by or they get scattered by the crystal, you still end up with the waves in phase. And so you end up with what's called constructive interference. And this will happen for all points in the crystal, as long as you're able to have the same angle and the same wavelength. And so you end up with something called a reflection or a Bragg peak. You will end up with some area where you have a lot of intensity. While for another random angle, actually, this is not exactly a random angle. This is a case where you have 1.5 periods extra for one path compared to the other. And in this case, we actually have complete destructive interference, because where one wave has the maximum, the other one has the minimum. And so when you combine them together, they will interfere and destroy each other, and you end up with nothing. But the interesting thing is that for most cases, even those where you don't have complete destructive interference, even if you have just some amount of destructive interference, because then you have not only two layers of atoms, but you have many layers of atoms, they will tend to cancel out each other almost completely. And only for those particular angles where you have constructive interference, you will see that you have not only constructive interference from the first two layers, but you'll have constructive interference from all the layers of the crystal, because everything will end up being an integer multiple of the number of wavelengths. And so we will end up then with this wonderful Bragg's law, which will tell you where you end up with your reflections or the bright spots in your diffraction pattern. So first we just observed the crystal as if you had multiple layers horizontally, but you can actually observe the crystal or view the crystal in other ways. So you can see it as also multiple layers in this other direction. And then, from Bragg's law, you're able, by knowing the location of the reflections and the wavelength of the light that you use, you're able to calculate the distance between the atoms, this d here, if you know both the wavelength and the angle. And this is just the order of reflection. You can have higher orders, 1, 2, 3, 4, and so on and so on. So this will give you one one distance between planes of atoms. But if you then look at other directions, you can get some other distance between planes of atoms. And you can look at the third direction and get the third distance between planes of atoms. So this will give you, you can obtain even a three-dimensional picture of where the different atoms are located. This is for at least very simple crystals. And this is just an example here. I don't know if this is going to work. Let's see. Yes, I think it will. So this is just in 3D. If you rotate a very basic crystal with a cubic cell in different orientations, you can see you kind of are able to see different planes in 3 dimensions. And you can examine, you can imagine each of these planes will give rise then to one of these diffraction points. So they finally managed to publish the first crystal structure, and this was from table salt or sodium chloride with this nice, very basic looking structure. And the reaction was a bit mixed. So while some of the people thought this was amazing, a great achievement, some other people, those maybe less, were not, they did not believe so much in this theory of X-ray and X-ray diffraction, were a bit skeptical, and they didn't think that this really looked like the structure of sodium chloride they were expecting. But eventually, it got more accepted. But this initial theory was only really able to tell you about rather simple crystals. So in the case where, so I represented here the atoms in a crystal, just as single atoms. But what about if you have more complex molecules making up a crystal? Then you need a little bit of more complex theory. And William Bragg, the son, was able to realize this rather early, actually. And he was able to make use of the Fourier theory, who had been developed quite some time ago for a completely different purpose, but he was able to see that using Fourier transforms, one would be able to analyze the diffraction pattern from crystals and try to read their structure. And I will not go into detail how this can be done, but this was realized at the time. And it took a rather long time to apply this to not natural crystals, because until then, all of the crystals from which the structure was determined were naturally occurring crystals, like the copper sulfate or sodium chloride. But people were then interested to try to look into more complex things, and things, for example, related to biology, because this is where we were trying to really understand things. So Dorothy Hodgkin was one of the pioneers here, and she was able to retrieve the structure of penicillin, which has this rather complex structure here on the bottom right, which you will definitely not going to be able to retrieve just by using the simple Bragg's law, and you will have to be able to do a lot of Fourier transforms, which at the time was rather tedious, as the computers were very limited, were mostly done by hand. And here you have on the top right the model of the electron density that she was able to retrieve for penicillin. But I think the most remarkable thing here was actually that she was able to get a crystal of penicillin at all, because it is totally not obvious that if you have a molecule that doesn't look like it will easily pack like this penicillin, somehow she was able to get a crystal out of it, and probably by first doing a lot of work to be able to purify enough penicillin, and then trying to create a supersaturated solution of penicillin, and able to get a nucleation point for penicillin, and then have an aggregation of more and more molecules. And somewhat miraculously, you don't get some amorphous thing out, but you actually can get a crystal if you hit the exact right conditions. And I don't think this was at all clear at the time that this was possible, but she showed that this was possible for penicillin. And I think it is rather miraculous that you can do crystals of such complicated molecules. Around the same time, Schrödinger, which you might be familiar from other things, wrote this book, What is Life?, where he was trying to bring together a more physical understanding of the phenomena of life. And one of the things that he theorized in this book was that it was already known that there was genetic material, in the sense there was genetic information in living things, because people had done a lot of tests, for example, looking at the transmissibility of traits in all kinds of plants and animals. So it was clear that there was some genetic information in living things. It was not at all clear how this was stored in people, and how this was propagated, and how this was mixed, and so on and so on. And he thought that this might be stored somehow in chemical bonds somewhere in us. And actually, only a couple of years later, there was this very famous picture here on the left-hand side. This is the photo 51 taken by Rosalind Franklin in 1952. And this is not a crystal diffraction pattern. This is actually a fiber diffraction pattern. So they had a fiber of DNA, and then they shine the X-rays on it, and you obtain this type of pattern. It's a bit different. You don't have the so clearly defined diffraction spots, but using also the theory from Bragg, they were able to analyze the different distances and come up with models. So in this case, it's not at all as easy to retrieve what is the origin, the molecule that gave rise to this. And in this case, it's actually the molecule is not even well defined, because as it was obvious, or as it was later learned, this DNA molecule is not a crystal, in the sense it will have different base pairs, and so depending on the type of base pair, you'll end up with different diffraction, but you will at least be able to get an idea of how the basic structure of things look like. And this, in just a year after, in 1953, James Watson and Francis Crick came up with this model, and also Maurice Wilkins came up with this model of the DNA based partially on this evidence from the X-ray diffraction, but actually also they said themselves they were inspired by the work from Schrödinger, that there was probably some chemical bonding information that kept the genetic information. And so that kind of showed how information can be kept in biological systems. And this was rather amazing, because it was seen that all of the living things had such type of genetic information. But going one step further, so we had complex molecules, but now we also people knew about proteins, they were curious about their structure. And proteins, it was also possible to crystallize them, and that is again quite spectacular. And you can see just here a collection of different types of protein crystals. And already in the 60s, Max Perutz was one of the pioneers of being able to crystallize hemoglobin, and he was able to obtain this wonderful model of hemoglobin, which is a relatively small protein, but it's much more complex than anything that had been done until then. So this was a great success. It was a relatively low resolution structure. Here on the right hand side, you have a modern structure of hemoglobin using a different type of representation. But it was clearly outstanding work, and he got the Nobel Prize for it. Here among the others, and the growth in the number of structures since then has been spectacular. So he got his initial structure in 60s, and the Protein Data Bank, the PDB, started to collect structures in 1976, and at the time there were only 13 structures in total, so not so many. And nowadays you have more than 100,000 structures are in total in the PDB, and about 10,000 new structures are deposited every year. So you have it's been an amazing growth in the total number of available structures. And this is just a small montage of the type of zoo of different proteins that we're now able to see at atomic level, and just because of, mostly because of X-ray crystallography. In the meantime, it was also realized that you could use electrons as waves and for imaging, and they will interfere just like photons. And they also have and they probably realized there was this relationship between the energy of the electron and the wavelength of the electron. And so you could get rather small wavelength electrons relatively easily. And this was already realized a long time ago. In the 1930s, the first electron transmission microscope was built by Ernst Ruska. And it took a rather long time, but nowadays, so this is a Nobel Prize from just a couple of years ago, it is possible to use electron microscopes to obtain extremely high resolution pictures of proteins and of viruses. So for example, here is just on the bottom left, a picture from a virus, and this is a single micrograph. And one of the main challenges was that if you put you have to put somehow you keep your proteins in solution, otherwise they will be not happy at all. But electrons, inside of the microscope, you have to have things in vacuum, so you can't have anything liquid. And one of the ideas is that, okay, then we try to freeze stuff. But if you just try to freeze your proteins, then the ice crystals that form will completely destroy your sample. And this was a conundrum for a rather long time. And actually, people thought this is impossible. It is impossible to obtain amorphous water that we can use then for to keep the ice crystals from destroying our sample. But Jacques Debouchet, he was the first one to be able to get vitreous water by very, very quickly freezing the very small amounts of water. And that then allowed to put the samples in the electron microscope and obtain the images that we now have. And just so it took about, I don't know, 80 years or so to go from having very, very crappy pictures from electron microscopy, which are, as you can see here, this is the picture of electron density from a protein. And on the left-hand side, you have actually the type of resolution you might have. Actually, even 10 years ago, it was pretty bad, or maybe 15 years ago. And on the right-hand side, you can see individual side chains, as you can see here on the bottom. So real atomic resolution. And this is actually from a single, from a single molecule in some sense. But these are typically averaged a couple of thousand particles. All right, and I think this is the end of the first part. And I think we'll have some time for break and questions. Or
yeah, so we have some questions about the first part.
The one who was the lady that we showed at the beginning.
Sorry, what?
I wonder who was the lady that we showed at the beginning with the blue eyes. Or it's just,
Oh,
just a Google photo.
it was a Google photo, I'm afraid. Sorry.
Okay. Other questions?
What does cryo mean in this?
So in the cryo part is that you want to keep the sample at a cryogenic temperature. So you of the, I did not go into the details about the ice, but to be able to keep things without having crystal ice in your sample, not only do you have to freeze the sample very quickly, you have to keep the sample very cold, because even if it's not like you can keep it just a little bit below zero. You have to keep it way below zero to prevent the phase change in the ice from going from amorphous ice to crystal ice. So the cryo comes from the cryogenic temperatures required. So it's typically done at liquid nitrogen temperatures.
So you have to freeze it like Han Solo, right?
Freeze it?
Freeze it like Han Solo.
Yes. 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 Without destroying. Yes. We don't.
And this picture that you showed, the last picture you showed,
This one here?
So on the right, is,
Yeah.
Is it achieved by microscope, or it's what we know it should be?
No, this is people nowadays achieve. So the typical distance between two atoms is on the order of 1 angstrom, and the resolutions that are achieved nowadays from electron microscopy is the best ones, a little bit above 1 angstrom. And this is done not, so it's not like you are able to obtain 1 angstrom from a single protein molecule, but what is done is that you have maybe about a couple of thousands, tens of thousands of those molecules in your sample, and then you average out the signal from all of these 20,000, and then this averaged signal will have a resolution up to, in the best cases, almost 1 angstrom. So really atomic scale.
So these dots are actually atoms.
So these atoms, even so, you can actually see maybe a bit here. This is probably kind of an electron density, this blobby thing, and the atoms are these ones are atoms, but this is a model. But what you retrieve is an electron density, which you then fit a model to.
So this is the question: where is model and where is actual?
So this is I think it's not. This was just to show as a demonstrate. It's just an image for. It's you will never have, for example, different types of resolutions, at least not this wide range of resolutions in the same type of sample. So it doesn't really show you the level of data that you might have. But this type of thing here, the more transparent looking thing, is more like the quality of the maps. So the electron density that you are able to directly retrieve from electron microscopy.
Okay.
And then to that, you will have to fit a model. And because you know that proteins are made of amino acids, and you know what is the chemical structure of the amino acids, there you have a limited amount of freedom to fit things to the electron density map.
Yeah. Other questions?
To my knowledge, some people also encapsulate this biological protein and other stuff in the graphene or other ultra-thin membrane.
Mm-hmm.
Having it in the liquid environment,
Yeah.
and insert it to the actual microscope. Are they able to get this quality of image?
So this using things in like graphene sandwiches is very, I would say, so doing it with the cryo method is relatively standard routine. Using things like graphene sandwiches is very much not routine, and it's an area of development. And yes, there is the hope that you might be able to do liquid phase electron microscopy, but to my knowledge, they are not able to get the resolutions that you are able when you're doing it like this. And I don't know exactly what is the difficulty. It might be that you end up with you need to be, the graphene needs to be extremely smooth and clean for you not to get a lot of extra background from the graphene, and also the amount of water that you have between the graphene layers has to be very small. Also, normally one of the difficulties when you're doing cryo-EM is that you want to have a relatively uniform and very thin layer of water, which is this is done on a grid which has some holes, and you have kind of the surface tension of the water will create a kind of thin membrane of water in the hole, and you want that membrane to be maybe 100 nanometers in thickness. So that is rather thin, and so all of these technicalities make it a little bit difficult to do things in liquid phase. But of course, that would be very interesting.
And this is a reproduced image from diffraction of the electron microscope, right?
No, I mean, typically they are not done in diffraction. You could also do it in the beginning. People did a lot in diffraction, and actually people did 2D crystallography with electrons. But nowadays, most of them are not done they're done in imaging mode, not in diffraction mode. So where you use the lenses to form an image and not go through the diffraction pattern.
Okay. So now we have break. During break, we will have experiments. So I brought a cathode tube, which you can see and try yourself. So it will be a real hands-on experience. The one that we showed at the beginning. So it's just outside now. And two interesting things. We had two lectures. One on statistical physics. their information and entropy. And Shannon entropy is work about information. It's 1948, 4 years after Schrödinger book What is Life. Early magnetic tapes to record computing, it was early 50s. And DNA came just after. So you have to kind of get an idea that people who are decoding DNA were inspired by Schrödinger book, and also people already know the idea of bit, like we use in computers these days. It's already on the market since Shannon works. So this is one interesting thing. We discussed this in lecture on statistical physics. Another interesting thing, we had lecture on Large Hadron Collider and high energy physics. And we started with Thomson experiment and Rutherford. And we have all this machinery which was used in biology, we of course also used in high energy physics. It was early accelerators. Cathode tube is the very first accelerator that people built, which we have just here. Now we have just slightly bigger one of size of outer loop around Stockholm. But it's the same, well, not the same. Well, actually I have magnet, so we will make it the same.
Actually, also for the, I didn't mention anything about the light sources, but a lot of some of the first uses or some of the people are trying to get better and better X-ray sources. And so they went to the particle physics community and they were able to use the waste X-rays that are coming out from their particle accelerators. This is in particular at SLAC, that they have a particle accelerator. So they basically just built a hole in the wall of the accelerator to get out the X-rays and then do experiments with the waste X-rays that the particle physicists really didn't like, because it just removes energy from their beam, and people then did crystallography with those kind of X-rays that came out. And then at some point they started to build light sources or synchrotrons, which are kind of like particle accelerators just to generate X-rays and not for doing research in particle physics.
Yeah, and all that you can hear on YouTube. We had lecture about this, right? So what he is telling, Philip was telling, we had lecture exactly about these things, about the Large Hadron Collider. So everything is very much connected. So let's go have some food and see experiment. So we come back at 7.
All right. So let's continue the final stretch. I just wanted to show you a couple of the there's the coronavirus. You might have heard about it, and you might have seen this kind of picture actually on the left-hand side. Is very often used in the media. And one of the questions that I had for me is how the hell do they know that it looks like that? And what are the parts, like what part from this thing is real and what is made up? And one thing that we know very well is this thing here in red. These are the so-called spike proteins, and they are the ones which interact with the receptors and are able to they take part in the process of infection. So have been extremely well studied. And here is just one example, one structure taken from this is a cryo-EM paper from the spike protein. So we do know that very well in high resolution for all kinds of different mutations and so on. We do know the spike protein back and forth, all kinds of extremely well, both by X-ray crystallography, by cryo-EM. People chopped off different parts of it. People did different mutations. People even did other kinds of many other kinds of methods, small angle X-ray scattering, which is a different type of method. So we do know that very well. In terms of the whole coronavirus, that is a little bit more tricky because the virus itself is not very reproducible in the sense it's not its structure is a bit flexible, and that is a real problem in the sense of if you're trying to obtain the whole structure of it, we cannot do it with a methods we currently have, at least to high resolution. You can do, for example, do something called electron tomography. So you can take a small amount of ice with a couple of coronavirus there, and you can try to take images with an electron microscope at different angles, and then try from those images to obtain the structure from the virus. But that will give you limited resolution, because whenever you try to image things, you also damage those things. For example, when you're doing electron microscope, the electrons will damage your sample, and that will limit the amount of resolution that you can get. And that's why you have to have many copies of the sample to be able to achieve vision. But the thing that you're trying to study is not, you're not able to get exact copies, and many of them, then you have a problem and you're not able to achieve high resolution. So different fragments of it, yes, we know. In terms of the whole thing, we kind of have low resolution knowledge, and that's why you can see in the picture on the left, some things are more detailed and other things are a bit more not put in such high detail, just because we don't really know so well, and it's more variable. So that's one example. So just want to show you here an example from a cover of a journal. Just thought it was funny that it also was seeing the invisible, even though I made that's the title before I saw this this journal cover. But I mean, it makes perfect sense. And what I just wanted to show you the type of illustrating how busy things are inside living cells. So this is just one illustration here. We have another one from the same artist. It's David Goodsell, who is a professor in Scripps in California. And here you have an image from E. coli. And you can see this is just one little bit of E. coli, and you see it's pretty busy. So you might typically imagine this proteins in a dilute solution or something. But in reality, things are very busy. But the interesting thing is that all of these cartoons, so these are cartoons, it's not something that is taken, it's not direct data, it's just an artist's impression. But what's amazing is that we actually do have structures for most of the things that exist in this picture individually, just not all together in the cell. And you can see, I think that you might be more familiar if you, for example, ribosomes, that they create proteins out of the RNA, they assemble the proteins, or you have here both the DNA and the DNA polymerase that's used to generate more RNA. So all of these machineries that we know how it works, when you put it all together, it gets really crowded and messy inside of a cell, and yet things still work. And as a final point, I have here just a video I wanted to show you. This is supposed to be an illustration of a human cell. Just to show all the, I don't know if I'm going to show you all the video, but the complexity of the different things that happen inside. And I think what's kind of amazing is that even though this is a video, made up video, but a lot of many of the details actually have bases on knowledge that was obtained from X-ray crystallography and more recently from electron microscopy. So a bunch of different processes of things that happen. So the amount of protein in these videos is a little bit too low. It should be a bit more crowded in reality than what they show here. But the idea is that the process that it depicts is rather realistic. So we have a bunch of DNA coiled inside. Here, I think it's showing how the DNA is coiled into with the histones to be able to compact that huge amount of DNA into something more manageable that you can actually fit inside of the nucleus of your cell. And again, all of these proteins that you see, they actually do have structures, just that, and we don't really know how they interact. The dynamics is still missing. So that's a big thing that we're missing in biology, is try to be able to see the dynamics of all of these things at atomic level. So right now we just mostly simulate these things. Yes, there's a YouTube link for if you wanna see, and there's more. And from this David Goodsell, he also has loads of different illustrations of like scientifically accurate illustrations of biological processes. I might not have sound. Oh yeah, for example, DNA replication, which is something that's very well studied and known, including all of the components of that take part in the replication. And it's kind of amazing how quickly they're able to open your DNA and split it in two and then make copies out of each of the halves. Yeah, and this is, like I said, it's real-time speed, so it's quite spectacular. And again, it's a bit one of the things that always has fascinated me about biology and this biochemistry in kind of some sense is that all of these enzymes, they do amazing things for any the chemists would say this is impossible. Now structures of all of these things, but we still don't really understand how these things work. So maybe if we, at some point, we might have real movies of how this stuff works and not just these model movies. Maybe we'll learn a little bit more of how this things work in practice. So yeah, now we have the DNA being transcribed into RNA. So we then eventually get the proteins out. Yeah, it's we are an amazing molecular machine. Yeah, I think if you want, you're welcome to. I can just leave it here, but this is pretty much the end. If you have any questions about any of this, you are more than welcome. Sorry? Yeah.
How can it be totally how can it be so fast to I mean, there are, I guess, the nucleotides, I mean, the bases around a GCP around, and they somehow they have a spot, I mean, is locked inside.
Exactly.
They place them
Yes.
So because they're like going in motion.
Yes. Yes.
Around and they fit.
Yes. I mean, there's they have this complementarity, right? So the right one has a higher chance of binding, but you have to imagine these are all very short distances and so on. So it's not strange that you are able to, on average, go this quickly. But in some sense, it's also still because you can have these small motions, these short motions, you can do it quite quickly. But because the amount of motion of the atoms is not in real time, you would expect the vibrations would be much faster in practice, right, in reality. It just the speed at which the DNA comes out matches the real speed. So there's a lot of diffusion happening and a lot of probing of the space, and sometimes eventually we're going to get the right nucleotide and so on to extend the chain. But this speed and so on, it's easy to measure by biochemical methods, just to see how large are the DNA pieces that you produce and how much of nucleotides you consume. But,
So in a sense, it's a Maxwell demo. Right?
yeah,
So I mean, like any configuration has approach, but there's also some good ones where
yeah, it is. I mean, there's this regarding the, for example, the case of the protein folding. There's this question of how, you know, how is it possible that it actually folds when there are so many different possible configurations that you have for all these atoms and so on. That was,
so do they have an answer, like do they like simulated like options that realize that this is only one, which is something you have? I mean, did you have to, so in a sense there are many options,
yeah.
and
People have simulated, for example, protein folding for small things, and they've managed to see that it does fold. But because this is done with force fields, so you have to use some kind of force field to, so you cannot do this quantum mechanically because it would take too long. So people use these molecular dynamic codes that are basically Newton's equations, and then they add a couple of extra terms to make it match a bit better reality. And for certain basic things, it kind of works. It still takes quite a long time, but you can't really do this for very large proteins or for this complex interaction, like a ribosome or a whole replication complex. This doesn't. No one has been able to simulate this in practice.
The main conclusion means we should abandon physics and switch to biophysics.
I think, yeah, biophysics it still has. There's still too many things that are unknown, but it is difficult to make progress because of all the things are so fragile and everything is so different, and yes, there are challenges, but it's all at the same time very interesting. Any other questions?
Question. So there was this part of the video where there was the double helix split in the
Yes, the replication.
some macromolecules I don't know, moving like a lever, like a robotic arm.
It was like this part of the movie.
Probably yes. So I was wondering what are these bluish molecules doing, because they move at a lower speed and they seems like just bending, going back and forth.
Which this part here?
Are they recomposing the helix?
They're going from a single strand to a double strand again. So this one kind of splits one splits the double helix into two single strands, and then the other one's a second strand from the single strands.
There are some videos where could more
yes, it's
maybe you can explain.
Yeah, I think it's a protein that walks on actin filaments. I don't know if I have.
Like, the one with some protein, and then there is this with 4 legs walking.
Yeah. Yes.
With this, exactly. They have some purpose,
Yeah, I don't. I think this might be related with muscle contraction. Okay, maybe not. Yes.
some questions.
Looks like I answered everything. So.
My dreams. My dreams physics.
Yes.
Yes.
Mhm. Yes.
I have a question. Okay, so they already know sequence of human DNA, right?
Yeah.
Did they find file structures, folders?
Not to my knowledge.
No, like, okay. Is there some like hierarchy of data, like how like it's organized?
Like I'm not an expert in the genomic part, but to my knowledge, no, there isn't really any hierarchical. I mean, even though there are different regions of DNA, definitely. For example, there are parts which code for proteins, parts which don't code for proteins. We have parts which actually come from viruses that then insert their DNA into our DNA, and all kinds of different parts of DNA. So it's not like it's uniform. So there are different regions, but it's not. I don't think there's a hierarchical
So there is
organization.
no hash table, right? There is no set of pointers to different.
Not that I know. No.
Okay. I mean, it's kind of clear that there is no, or people just do not know.
I mean, it has. If there is, it might be that we're not smart enough to understand the organization. I don't think there is a way to disprove that there is an organization, but it's not apparent. I mean, there is some organization in the sense that sometimes you might have, and might be, and might have some. For example, you might want to express some genes together with some other genes, and you have to put them close to each other. So you might have the same promoter to express both genes at the same time. So, but that's not a very hierarchical. That's only like maybe one level, very basic level of organization. Not something like your folders, especially not recursive organization.
Okay. And why? So do people understand a little bit why it's base 4, not base 2 computation?
Well. No, I would not say that. I mean, you might. One kind of cheating answer would be that we have. So the ribosomes have 3 spaces for you to put your genetic code to code for one amino acid. So we have 20 amino acids naturally occurring in our proteins, and if you only had 3 bits for it, you wouldn't be able to code for all of the different 20 amino acids. But if you use something which has 4 values, then you're able to using 3 bits or three slots or 3.
Okay, so 3 slots of DNA is one amino acid.
Yes.
Right. I see. So
but then we might say, okay, but why don't you have a ribosome which has more slots than? But that is the reason why, and so there's a redundancy in the genetic code. So it's that you might have multiple things coding for the same amino acids. This happens, and you also have some that code, for example, for the beginning and for the end. Actually, yeah, for the end of not so much for the beginning, but for the end of the protein.
How many letters?
3, all 3,
right?
yeah, always 3.
So one.
No, because I mean, you only have 3 spots, so it cannot be more than 3. And you might say that, okay, if you have more spots, it becomes more complex. The bigger the protein, you have to waste more energy to make those. And so on.
Okay.
Because you have a lot of ribosomes in your cell, a lot of energy has to be spent on making those. So if you make them double the size, for example, then you might spend double the energy, and you might not want that.
Okay, so it's also 332 configurations, 25 amino bases, a little bit. Okay. It's more, it's 64.
Oh, with the 4 times 4 times 4.
Okay. So any other questions? Okay, so let us thank Philip.
Thank you very much. Oh, I should put. Wait,
Right. So
wait,
one
yes.
last,
Yes, one last. I forgot.
one last thing from me.
There is someone in the chat thanking everybody for the series of lectures, especially for what you're gonna say now, probably support.
Yeah. Thank you. Right. So this is the last lectures that we had in this program. It is so far. So thank you for coming. And I would like to say that these lectures were organized to raise funds to support Ukrainian people. So all the lectures are available online, and there is links to YouTube, and there are also slides for them, so you can watch more if you want to learn more. Myself, I learned a lot myself by preparing. I finally find the time to learn physics. I mean, seriously, like much more than I learned before. I learn now, not just computing integrals, just doing real physics. And yeah, so we try to assemble all this we learned in the slides. You're welcome to come to the website. And if you support the cause, please donate some funds and leave us small feedback that you did it, so we know that it actually was useful. Out of all registrants, we have 10% who actually said that they did something. It doesn't mean that nobody else did something, but there is also like part of the story that you come back and click and say, okay, we did something, so we know. It's completely anonymous, it's up to you, but please do it if you want to help and let us know. So thank you for coming, thank you for your interest, and, well, have a nice summer.
So we should thank Dmitry for organizing this series of lectures. It was able to work.
I think we should also thank you, actually, for setting up all these series of lectures.
Yeah.